Hello friends, in a previous lecture we already discussed about diversity and microbial habitat of microbial extremophiles. Now today we will discuss about sample collection, enrichment, isolation, classification and properties as well as their application and adaptation of alkaliphiles. Now we will start it. Alkaliphiles. Alkaliphiles are a class of extremophilic microorganisms that capable of survival in a alkaline environments, growing optimally around a pH of 10. Alkaliphiles includes prokaryotes, eukaryotes, and archaea. These bacteria can be further categorized as obligate alkaliphiles, those that require high pH to survive. Next, facultative alkaliphiles, those able to survive in high pH but also grow under normal conditions. And haloalkaliphiles, those that requires high salt content to survive. Then alkaliphilic microorganisms coexist with neutrophilic microorganisms as well as occupying specific extreme environments in our nature. Microbial growth in alkaline conditions presents several complications to normal biochemical activity and reproduction as high pH is determinantal to normal cellular processes. The sample collection of alkaliphiles. To collect the sample of alkaliphiles, you can start by identifying alkaline environments such as alkaline lakes, soda lakes, alkaline soils, or alkaline hydrothermal vents. Once you have identified a suitable location, you can collect it. Samples using sterile sampling tools such as scoops or corers. Make sure to maintain aseptic condition to avoid contamination. Store the sample in a sterile containers and transport them to the laboratory for further analysis. Next, enrichment of alkaliphile. In enrichment, to enrichment for alkaliphiles, you can use selective media that mimic alkaline conditions. One commonly used medium is an alkaline broth or agar supplemented with alkaline salts and a carbon source. This creates an environment that favors the growth of alkaliphilic microorganisms. This incubate the enriched culture at the appropriate temperature and pH for alkaliphiles. After incubation, you can isolate and study the alkaliphilic microorganisms from the enriched culture. In isolation of alkaliphiles, use ELB agar media with pH 10, serially diluted soil sample and spread them on pH 10 agar plates. Incubate overnight at 37 degrees Celsius and next collect and purify alkaliphilic bacterial colonies. Then classification of alkaliphiles. Alkaliphiles are microorganisms that requires an alkaline pH for growth and survival. They can be classified into three groups. One is obligate alkaliphiles, second one is facultative alkaliphile, and third one is haloalkaliphiles. So obligate alkaliphiles are the required high pH. Then second one, facultative alkaliphiles require they can survive in both alkaline and normal conditions. And third one, which is haloalkaliphiles, which require high salt content for survival. Next, adaptation of alkaliphiles. There are molecular adaptations. Alkaliphiles contains unusual either lipids bonded with glycerol phosphates just like other archaea. In this case, lipids long chain branched hydrocarbons either of phytanely or biophytanely type are present. The intracellular pH remains neutral in order to prevent alkali liable micromolecules in the cell. Then alkaliphiles have developed various adaptation mechanisms to survive in alkaline environments. One key mechanism is the reproduction of alkaline stable protein and enzymes. These molecules have unique amino acid compositions and structure that allow them to function optimally at high pH level. Alkaliphiles also have a specialized transfer system that helps regulate the internal pH of their cells, maintaining a balanced environment. This adaptation mechanism enable alkaliphiles to carry out essential cellular processes and thrives in a alkaline condition. So next, last topic is application of alkaliphiles. The alkaliphiles promise several interesting uses for biotechnology and future research. Then, alkaliphilic methods of regulating pH and producing ATP are of interest in a scientific community. However, perhaps the greatest area of an interest from alkaliphiles lies in their enzymes like alkaline proteases, starch degrading enzymes, cellulases, lipases, xylinases, pectinases, chitinases, and their metabolites, including 2-phenylamine, carotenoids, siderophores, cholic acid, derivatives, and organic acid. Next, it is hoped that further research into alkaliphilic enzyme will allow scientists to harvest alkaliphiles enzymes for use in basic conditions. Then next one is research aimed at discovering alkaliphiles produce antibiotics shows some success. It has been held at day by day by the fact that some products produced at high pH are suitable and unusable at a physiological pH range. Thank you. Next topic we will study in next lecture.